So good morning, everyone. Welcome to the invited session 15. The title of our session is Complex Response Dimension Reduction and the Data Integration with Application on Neuroimaging. I'm Zhao from Yale, and I'm the session organizer and chair. Today, we are very honored to have three excellent speakers, Dr. Xi Luo, Dr. Liu Duan, and Dr. Kristin Linn. So each speaker will have 20 minutes, and depending on the time, we may have like time to answer one question or leave all the question to the floor discussion. But during the talk, the audience feel free to just type the question in the chat. So let's start with our first speaker, Dr. Xi Luo. She is from the University of Texas, and his talk today is about covariant assisted principal regression for covariance matrix outcomes with an application for fMRI. Let's welcome him. So Xi, feel free to share the screen.
Yeah, thanks a lot, Yize, for the invitation. And thanks a lot for organizing this session. It's really glad to talk at SMI. This is one of my favorite conferences. And let me start sharing my. Okay. Can you see my screen?
Yes.
Okay. Today I want to talk about some recent work with my collaborators. So on a methodology so-called CAP, and so and here are the co-authors, and this is a joint work with Brian Caffel. I just learned that Brian is talking on the other session on some related methods as well. So if I had known that, I probably would choose something else to talk. But this is also done by my former student, now assistant professor at Indiana University, Zhao. And she does a lot of work, and she's the first author of the paper, and also joining with student Bing-Kai Wang from Johns Hopkins, and Stuart is our clinical collaborator. And you can also view the slides on this link, bit.ly SMI Cap 21. So what I want to make this talk in 20 minutes is really trying to motivate and cover a lot on the model part. And we did have some theory and also scientific applications, but I will not choose to focus on that. So let me briefly discuss a bit what do we mean, what we are working on, what kind of data we're working on. And we're working on fMRI data. And here is a, we're working on specifically resting state fMRI, basically subjects lying in a scanner and do nothing, I put in quote, but the subjects could still have lots of, thought processes going on as well. And it is very interesting for neuroscientists and many scientists to study so-called brain networks and to study how different brain regions, if you look at the lower right corner, how the different brain regions are interacting with each other. If you look at their time series showing us those cutout boxes. And so one standard approach to look at brain region connection is to examine the correlations of those time series from different brain regions, and that have been widely used. And so brain networks, and I think in this conference, you probably also heard many other excellent talks on brain networks. And this is a really emerging trend in research. And in the past, there are a lot of research on estimating brain networks from data. And we contribute a bit to that part of work, mostly on inverse covariance matrices, as well as on so-called DCM. In the past, we also look at those networks and trying to figure out how the information flow in those networks, or try to use those networks for clustering brain regions. But then so we think a lot about those approaches, and then we think is what else can we do with those brain networks? So in this talk, we are trying to think about how to utilize or how to link those networks to other important variables. And so that's the data integration part we are trying to address. And specifically, we're trying to address, explain the network differences. And so let me give you a motivating example. What we have here are 3 subjects, and they obviously have different age and sex, and because of that, they also have different brain networks. And so it is important neuroscientists and want to know is that why the brain networks are different, and can those differences in brain networks explained by those covariates like age and sex. And obviously our work as a statistical measure can be extended to cover many other covariates as well. So put it simply, we just want to know how the covariates will impact those network connections. And put it mathematically, we want to take a graph and write a function, and this is looks like already like a familiar generalized linear model, an inc function of the graph, and I'm going to equal the covariates times different beta coefficients. And at this, if we could achieve get a model like this, and the scientists could interpret, say, the beta coefficient associated with age will impact the brain networks as such and such. And so that's the goal of our method. And if you think this mathematically, and because we don't usually directly observe the networks, we have covariance matrices as one way of modeling the brain networks or estimating the brain networks. And we are given n semi-positive definite matrices, and I call it sigma I here. And we're also having different vector covariates, age, sex, and it could have many other variables like DSM-5 and various cognitive scores and etc. So what we want to do is really just find this function such that, and for each matrix, and we can build this model. And this turns out, this already looks familiar like a regression problem. And the complication here is that the outcome is the sigma I, is the P by P matrix. And in essence, we just want to regress positive definite, and the population version will be positive definite matrices on vectors. And some related problems, working on covariances has been done in the statistical literature, starting in the time series or in economic metrics, and they trying to model the a scalar variance. And for covariance models, a lot of excellent work on that. But what they're trying to model is not doing dimension reduction on the covariance matrix per se, but they just want to take the vector covariates and make them symmetric, make a symmetric matrix, and using that to approximate the sigma I as shown here. That's what their approach. So we are trying to put the link function on the left side so that we can trying to do dimension reduction of the covariance matrix, and that becomes advantageous when we later talk about some recent extensions of modeling extremely high dimensional covariance matrices. And there is also multi-group PCA, and the goal there is really trying to compare different covariance matrices from different groups. And so put it in a regression framework that is about comparing when you're trying to handle categorical variables indicating different groups, but they cannot handle, for example, continuous, categorical, and also even interactions and etc. And lastly, not the least, and they also have tensor on scale regressions as a very general statistic framework, but they usually don't guarantee positive definite matrix outcomes. And so neuroscientists have thought about this problem. The method they come up with is sort of very simple, and they just take, say, if I have a covariance matrix, how about I just take each entry of this covariance matrix and do a regression? This is a so-called massive edgewise regression. So they're not treating the outcome covariance matrices as a matrix, but rather they just treat it as a connection of individual entries. And what they propose to do is for the p-square number of entries, but if you count the symmetry, you're probably gonna discount that by half. And they just take almost the p-square order number of regressions, and at the end of the day, they also recognize there is a multiple testing problem because you're testing on p-square entries, and so you have to do multiple corrections like FDR and etc. But once they do FDR correction and like that, you realize because you're doing it on the order of p-square, you are sacrificing a lot of statistical power, and because the number of tests is really large. I mean, if you think about p could be hundreds or even thousands, and you also those multiple testing correction procedures, there are some development trying to account for dependencies between those regressions, and but put in short is that usually the neuroscientists they don't know how to deal with that. So what we aim to do in a thinkless way is that we want to do some kind of PCA type approach on the covariance matrices, and then I can use my covariate xi to explain my sort of like principal components. And however, what we realize is that we should not just directly do it in two steps, like say first is to do a PCA, second step is to do a regression on those principal scores, or I always mix up the loadings or scores. And so we should not always do things like that, because I have some example I'll show you in a moment. And this is a little bit different from other PCA methods, and they usually use PCA on the predictors instead of on the outcome, and then they call it supervised PCA or some regression, so that they can reduce the dimensionality of X that has been well studied in the statistical literature as well. So we have other extensions. We get more time, or talk probably one or two extensions of this work. So let me start with this toy example. And what we are trying to find here is we have a covariance matrix sigma I, and what we are trying to find is we're going to call principal direction. This is different from principal component projections, and for the moment, I'll hope I will make that clear. So this gamma, multiplying this sigma I from both sides, and then we're going to take log of that, and this is if the gammas are eigenvalues of this sigma I matrix, and this will be the log of the eigenvalues. And we're gonna, this is our link function on the left side, and the right side is just the regular regression formula. And how on our PD, our principal direction is different from principal component, mainly because that our direction PD is not necessarily the eigenvectors of the covariance matrices, but rather we are trying to seek the direction such that has explained the highest variation related to X. If you look at this this picture right here, and we have different ellipsoids and representing covariance matrices in lower dimensions like 2, and the different color grayscale representing the corresponding X values. So you can think is if we can generate covariance matrices depending on different X values, and here we visualize them. As you can see, if you do a principal component, and the direction you will find, or by the eigenvector, is this 45-degree line from lower left to upper right. And because this explains the largest variation along that direction. However, if you look at along that direction, it doesn't really, it is not really the direction that explains the most variation in X. So, but rather we want to go the 90-degree orthogonal to that, and that's the direction our method will find. So by this red one from upper left to lower right. And for the interest of time, I will skip some of those. And let me just get into the measure a little bit more quickly. So put it away is that we are doing some maximum likelihood approach. However, we cannot directly apply that. We have to consider a constraint, because otherwise the solution will be really trivial, like gamma equals 0 or something. And so, and the constraint here, we spend a lot of time thinking about this constraint. And one obvious one is just to take this H to be identity matrix, and this will be corresponds to the L2 norm of gamma. However, that does not really work, and because we have a theorem proving that, so what we are advertising here is that you should take the average of the sample covariance matrix and use that as the H matrix. And the reason we cannot use the identity matrix in the previous optimization problem is that because if you use that, the solution is really corresponds to the minimum eigenvalue of the matrix, because we're in fMRI and also many scientific fields that we think like those small eigenvalues or those principal components of higher orders, they're usually related to the noise. So that's why we thought about this H equals identity first, but it does not really work. And algorithm part seems to be straightforward given the advances in the past several decades on optimization algorithms. So we just did iterative algorithms to update both beta and gamma. And because we want to estimate the regression coefficients beta as well as the principal direction gamma jointly, so we're gonna just iteratively update them. And some theoretical results, and we can get the consistency of our coefficient estimate, and as well as we can get when we estimate this gamma from the maximum likelihood estimator of using the Florey, who studied the population or multiple principal component problem. We use that, and then we can also get, if we use that, we can also get the consistency and convergence rate for our beta estimates. And I'm almost running out of time, so I'll be quick on the simulations and the real data set. On this simulation study, in the paper, we consider other scenarios, and put it a simple way is that here we're trying to compare our approach with the PCA, called PCA here, as well as the common principal component analysis developed by Florey, and which is also we build our theory a lot on his work. And here you can see is that the first direction to choose is -1, and using our approach, we're gonna find the -1 by the first direction, because again, our methods trying to look for the direction is associated with the covariate X. But if you do PCA or common PCA or other principal component approach, they will not find the first component is not getting a regression coefficient -1. They turns out to find some directions is even irrelevant, almost irrelevant to X, because you can see the regression coefficients are almost 0 here. And in the fMRI application, we apply to a data set from HCP, and we compare with the so-called massive edge regression case. And it turns out if you do those massive edgewise regression or running p-square regressions, you will not find statistically significant results. And by using our approach, we can find several components here shown by the 95 confidence intervals. You can see that for several components on the y-axis, D1 to D5, 5 components, and some of them are directly associated with age and sex, and 3 of them are associated with the interactions. And lastly, we just want to briefly mention is that for this regression framework, we can also take the gamma and visualize it on the brain and see what are the regions that we pick, and so that we can pick from the covariance matrix, as well as we map that back to the brain regions. What are the regions that we can take, and that those regions will contribute to the link function, such that the link function is can be explained by the covariates. And here are some of the regions that discover, and the scientists seems these regions make sense. And probably just one few seconds to explain, we have some work on extending this to high-dimensional covariance case for voxel level connectivity matrix already published, and we have an intermediate case that it's already under revision. And so there we have to do a little bit of work on the covariance matrices, so we cannot just directly use the sample covariance matrix. And hope I will finish on time. And today's talk is really just about thinking about how to regress positive definite matrices on vectors, and we're trying to use that method to explain how brain networks are different between subjects using fMRI, have some theoretical justifications, and we actually also have a package available. And that's the end of my talk. Thank you so much for your attention. And any comments, questions?
Our next speaker is Duan from University of Florida. His talk is about the Bayesian Vector Autoregression using the tree rank prior with application to fMRI analysis. So Leo, is all yours.
Okay, thanks, Yize. Maybe I should shorten my title a little bit. It's a little bit long to read. So let me share my screen here. Okay, can you see my screen right now?
Yes.
Okay, let's see. So let me switch this to presentation. How about now?
Works fine.
Okay. So I'd like to talk about this thing called the vector autoregression, and just to clarify, this is related to graph estimation, and this is also motivated by analyzing the resting states fMRI data as she introduced early. So, but what we're going to see that we are trying to estimate a different type of graph that is called the Granger Causality. So this is a little bit different from the covariance. Covariance estimation based graph estimate. Okay, so so this is a joint work with my graduate student Zhao, and also George Machalidis and Zhengwu Zhang, who's also in the audience right now. Okay, so if you have any questions, you can also direct your question to him. Okay, all right. So here's just the outline of today's talk. So first, some background on this vector autoregression, and also what's the Granger causality graph. And then I will switch to introducing my proposal. This is a new type of regularity that is based on the graph topology instead of the matrix norm. Okay, and then I will show you some combinatorial theory as well as some statistical theory related to this model, and then we're going to see the results applying this method on the RS fMRI. Okay, so just to give you some background on this vector autoregression, usually use the acronym VAR. So what it does is that if you think about a multivariate time series, such as fMRI video imaging, then we have p-dimensional. So if we have p sensors and then we have a p-dimensional outcome, right? So we just collect them over time, over discrete time, from 1 to capital T. And then we can think about the current value of yt as a linear combination of the past few d time intervals, and then we just do a linear combination, sum them up together, and plus that epsilon term. Okay, so this framework is called a vector autoregression as a multivariate extension of the AR process. Okay, so what's interesting is that this type of data, I mean, this kind of model is very high dimensional because, as you can imagine, we are dealing with basically vector outcome, and each of this C is a R by R matrix. So we have altogether we have P times D many matrices to estimate. So this is a very high dimensional model, and so in order to have a successful signal recovery, we need to apply some assumption, such as sparsity. So if we have a sparse estimate of C, very interestingly, this is related to something related to this thing called Granger Causality graph. Okay, so you may wonder, how is this different from, let's say, the very commonly used Gaussian graphic model? Okay, the key difference is that if we look at this epsilon term, what we can do is that we can actually assume this epsilon to be a correlated noise. So this means that we can estimate the covariance of this and explicitly exclude it. So meaning that we can discard that part of the covariance from our total cross covariance estimation. This is because usually, as you can imagine, when we are using different equipments, there are some background noise that may be correlated. For example, you could have, let's say, the brightness of the image from 1 second may be brighter overall, and the next second maybe the picture may get darker. So in that case, there are lots of spurious correlations there. And by explicitly model their correlation and exclude it later, we can perhaps find something more meaningful other than, say, just the change in the brightness of the picture. Okay, so as you can imagine, if we can just rely on the estimate of those C's, we can build this thing called the Granger Causality. So what it basically shows you is that there is some influence of the past observation to the future observation. Okay, so just for the purpose of graph estimation, so if you have a certain Cij that is not equal to 0, this means that there is a dependency of yi on the yi on the yj from the past. Okay, so in that case, we say the outcome yj is Granger causal to yi. Okay, so I should clarify that this is not exactly equivalent to the true causality. However, this can be served as a meaningful starting point to identify some true causality. Okay, so this vector autoregression model actually has a long history. So because of this graph interpretation, sort of in the causal flavor, and also usually these models, they enjoy very good predictive performance. So combining these two, this has led to a very wide popularity even outside of the statistics. For example, I think this thing has an origin in econometrics, as well as politics science, and most recently, it gets very popularized in the neuroscience. And Minzhou Ding is one of my collaborators, and he's the major leading figures in that field. So basically, this thing become very popular in analyzing the functional connectivity. And of course, in our field, there has been a very rich statistical literature on that as well. Okay, so just to give you an incomplete survey on those methods, like I mentioned earlier, this is a very high-dimensional problem. So as you can imagine, naturally, L1 norm-based sparsity method has been applied on this model. So there is just applying L11 norm regularization or something like group lasso, and a particular paper worth mentioning is Basu and Machadidis on the annals of statistics, establishing important theories related to the sparsity regularized VAR model. Okay, and of course you can also consider some low rank structure in this vector, in those coefficient matrices, and perhaps the most related method to our method is something called the Minnesota priors. Okay, so I guess this got this name because of this Bayesian shrinkage prior, those Bayesian shrinkage prior on the VAR model were first proposed by several professors in from the University of Minnesota. And so what they basically say is that we can put some informative Gaussian prior on where those coefficients will likely to happen. Okay, and like I said, this is very likely to be an incomplete list, and there are many other existing work. Okay, so that just leads us to a question: why bother building another VAR model? Okay, so when I initially started looking at this problem, I tried some of the existing methods. For example, I tried the lasso regularized VAR model, and it fits the data perfectly. Okay, and when even doing cross validation, when doing out of sample prediction, the performance was excellent. Okay, so why are we making another VAR model? Okay, so in this case, we are basically motivated by getting a better interpretation. Okay, so here's the results. Here's the preliminary result. Like I said, I got when initially fits our rsfMRI data using the lasso-based model. So as you can see, so here I'm using the DESIC LS, which consists of 68 nodes. So basically here's the left hemisphere of the brain, and here's the right hemisphere of the brain. So as you can see, this is what lasso told us. So this is where the Granger Causality that the lasso thinks. Okay, and on the right hand side, this is basically the same graph except just using different layout. Okay, so as you can see, lasso did tells several edges. That seems very important. Okay, and you can see that there are many isolated nodes. Okay, but when I initially show this result, well, I guess when I initially show a different graph related to EEG to my collaborator, and their response was, this looks nice, but shouldn't the brain be a connected network instead of one with many isolated components? Okay, so as you can see, we get some incoherence between statistical finding and what neuroscientists believe to be true. Okay, so again, so as you can see, as we've seen many times in this field of machine learning and statistics, sometimes a very predictive model does not mean that it is interpretable, or does not mean that it is necessarily being plausible to some domain scientist. Okay, so that means there is still a chance for us to build some improved model, at least improve in the sense of being more interpretable. Okay, so now let's just re-examine this model, just using some simple linear algebra. If we write this in the matrix form, as you can see in this high-dimensional framework, if we can write this autoregression in this sort of like a big Y equal to X beta kind of form, you can see that the columns in this big X matrix, they are highly correlated. Okay, so that means lasso isn't the only best solution. There are many multiple equivalent solutions. So meaning that instead of using lasso, we could build some other alternative model. Perhaps that will give us some equally competitive predictive performance while being much more interpretable, or at least be more plausible to your scientists. Okay, so this is the goal of this project. Okay, so the key is, so I guess the fundamental question is, how do we ensure or at least encourage connectivity instead of getting many isolated components? Okay, so let's just start from the basic. What is the simplest connected graph? Okay, so actually this is related to a very famous computer science concept called the spanning tree. Okay, so basically the spanning tree is a graph, is the smallest connected graph. So meaning that if you have p many nodes and you are only allowed to use p minus 1 edges and you want to connect all the nodes together. Okay, so this concept has the origin of solving the traveling salesperson's problem, meaning that if you imagine being a salesperson and you want to travel around p many cities, you want to minimize the total distances that you need to cover in order to travel to all of the cities. Okay, so solving this problem will give you something called the minimum spanning tree. Okay, so here's an illustration. Okay, so here is the edges to represent the possible roads, and so here gives you the minimum spanning tree on which you can build an itinerary to travel to all of the nodes here. Okay, so as you can see, we have the smallest connective graph, and how do we use this to build a VAR model? Okay, I mean, how do we use this to regularize the complexity of the graph? Okay, while preventing it being too restrictive. Well, the idea is that we can borrow the concept from the metric entropy of covering. So let's just imagine, suppose we have a graph. I mean, it can be connected or disconnected. I mean, there could be some edges that is lost here, but here what I'm showing you is a connected graph. So for any graph, we can imagine cover this graph with several numbers of spanning trees. Okay, so here shows you covering this graph using 2 spanning trees. So here's the red is one of the spanning trees, and the bluish color is another spanning trees. So we can cover this graph with 2 spanning trees. Okay, so we have some elementary derivation showing that actually for any graph you can cover the graph with p minus 1 spanning trees. So meaning that we have something similar to the idea of graph. To the idea of the rank, except the matrix rank is something defined regarding matrices, but here we are defining a new rank related to a graph. Okay, so we have a complexity measure, which is the smallest covering number of a graph, and we call this the tree rank. Okay, so what's interesting is that when you have a small tree rank, of course you have a graph with low complexity, so we can use that to encourage a simpler graph while encourage it to be connected. But at the same time, as you will notice, when you have a low tree rank, you will automatically get sparsity. Okay, but the reverse is not necessarily true. Okay, and why is this useful for a vector autoregression? If you think about it, when we are considering a cardinality restricted model, meaning that we just restricted the number of non-zeros that can appear in our coefficient matrix, and if we just force these non-zeros can only appear on the connected graph, then we will very likely to get a connected graph. Because in order to fit the data well, the model will automatically give you a lot of non-zeros, and this is where the low tree rank kick in. So basically, it will just encourage those non-zero to appear on the to form a connected graph together. Okay, so here's the details of our prior. So basically, we take a normal scale. Hierarchical model. So here, each A is the adjacency of the spanning tree, and we just sum them together. And we use a dirichlet prior. Basically, this S will be sparse, and we can set a arbitrarily large M, so that some of the SL, when it become close to 0, it automatically just reduces the number of effective trees that we need there. Okay, so and I guess I will just skip some of the details, but what I want to say is that, as you can imagine, as long as any of the scale is close to 0, then this Cij will be close to 0. Okay, so you may say this looks nice conceptually, but how do we pull this off? Okay, so as a matter of fact, this is a very challenging combinatorial problems, because a graph can have as many as p to the p minus 2 many spanning trees. So this combinatorial space is actually huge. The complexity is very high. Okay, so if you talk to a computer scientist, usually he or she will tell you that in order to make sure that you are estimating a spanning tree, you need to do something called the graph traversal, meaning that we start from one node and we go to the next node and make sure that they are connected. So as you can see, this is a very burdensome, cumbersome process. Okay, so we need a much more efficient way to bypass this. Okay, so here's just some combinatorial theories that we discover, and personally I believe that this is very, very interesting, because it just allows us to directly bypass this huge combinatorial place, and we can obtain some very nice closed form result. Okay, so here's some details. If we consider a spanning tree randomly generated based on the weighted matrix, okay, and we assign this probability as this. So basically, this is something similar to a Gibbs distribution, and with a probability proportional to the exponentiated to this weight, and then we just multiplied it over the edges over the tree, and we have this normalizing constant that's usually known as the partition function. And as you can see, this summation will basically, this summation happens in a huge combinatorial place, as I mentioned. However, what's interesting is that we can in fact compute this partition function in the closed form using the simple matrix determinant. Okay, and why is that true? Okay, so here's the diagram. Okay, so this is basically an extension of this thing called the Kirchhoff matrix tree theorem. What it shows you is that here's a diagram of how we calculate a determinant, right? So if you think about how we calculate determinant, what it basically does is that we do a summation of all the product, where each product is that we just visit each row and each column once. Okay, does that sound familiar? When we visit each column and each row once, it's similar to visiting each city for only one time. Okay, so this is the link between this summation and the determinant. Okay, so this means that we can, in fact, just compute this normalizing constant by just computing a determinant of a weighted Laplacian matrix. Okay, so now if I just take a log of this z and take a derivative, and what you will see is that it will basically give us a closed form solution for estimating the probability for a spanning tree to cover an edge, to include that edge ij. Okay, so now you may notice that I have another working parameter tau here, and what now if we let this tau go to 0, you'll see that this probability matrix with each element between 0 and 1 will actually converge to being very close to either 0 or 1, meaning that this will converge to the adjacency matrix of the maximum spanning tree. Okay, so what this gave us is basically give us a continuous relaxation for the spanning tree. So not only this is continuous, but this is differentiable. So meaning that we can put this inside of the normal scale hierarchical model, and then we can just run Hamiltonian Monte Carlo for posterior estimation. Okay, so I guess just for the sake of time, I will just skip some of the statistical theory results. So basically, we develop some simplified results for the stability results, stability condition for the graph-restricted VAR model, as well as the posterior concentration rate. Okay. So I'd like to show you some of the results applying this framework on the resting state fMRI data. And so here's just a show of the raw data, and here's the results that we obtain from our model. So here I'm showing you the posterior mode. And so in the posterior mode, we found 3 dominating spanning trees. So here I'm just showing you the first 2. So again, just to remind you, this is left, this is right hemisphere of the brain. So as you can see, the first tree corresponds to basically the symmetry between 2 hemispheres of the brain. Okay, while the second tree, you can see that we have some more local connectivity. Okay, and again, just to show you, this has a very good fit to the data. So as you can see, so the blue has the raw data, and the fitted, they're very close to each other. So as a matter of fact, compute the R-squared. R-squared is like above 99%. Okay. So again, I should clarify that so if you use some other methods, regardless of this method, such as lasso or elastinet, you will get similar results. But I believe that we just have something that is more interpretable. Okay. And as I mentioned earlier, so what this gives us is that, or this VAR framework gives us, is that we can explicitly estimate and exclude the zero slack correlation. So a lot of these correlations, they could be just due to some, they just could be spurious in nature. Okay, so just to show you some comparison of the alternative. So previously I've shown you how using lasso. While if you use the elastinets, well, certainly you can get many edges, but in this case, we have too many edges if we just use the elastinet. Okay, so really, if the tree-based regularization give us a middle ground. So basically, we can use as fewer edges as possible, but while still encourage the graph to be connected. Okay, so that's all for my talk. Just to briefly summarize. So what I show you today is that we can actually use spanning tree as the building blocks to regularize our model based on the graph topology instead of just using matrix norm. And I've shown you that this combinatorial constraints, they are very useful, but they can be a headache for estimation. But using some combinatorial theory, we can completely bypass the combinatorial search, and we can build some very interesting model while still guarantee that the posterior is computationally tractable. Okay, so here's the reference. I should say a lot of the credit should go to my collaborator Jing Wu, and also my PhD students. She does most of the work here. So Yuan, and I guess that's it. I'll be happy to answer any question at the end of this session. All right, thank you.
Our final speaker is Kristin Linn from UPenn. Her talk is Inter-modal coupling for multi-modal imaging analysis.
Great. Can you see my slides?
Yes.
Great. So thank you so much, Yize, for the invitation to speak in this session. I'm really excited to talk today about a framework that our group has been developing for studying relationships between different imaging modalities. And I just want to start by acknowledging all of the people that have contributed to this work, including several students and colleagues at Penn and in other places. There's a lot of people that I couldn't fit their pictures on this slide, but I'm really thankful to everyone who has contributed to this work. So here's a brief outline for my talk today. I'm going to give some background and motivation for the types of questions that we want to study and the framework that we've been developing. And then I'll spend some time giving you a description of the intermodal coupling model and our estimation procedure that we've been using. So we're referring to this method as intermodal coupling, or IMCO for short, so I might use those terms interchangeably throughout this talk. And then I really just want to focus most of my time on telling you about some exciting applications that we've been using IMCO for, in particular, using this framework to help segment lesions in the brain that are caused by multiple sclerosis, as well as correcting for partial volume effects and studying questions of interest in neurodevelopment. So I don't think I really need to sell you all on the advantages of multimodal imaging, but I do just want to briefly point out this great figure from a review paper that appeared in Brain Informatics in 2015 on multimodal data, where they really outline sort of the advantages of each different type of modality for various aspects that we might want to study about the brain. So on the left column here, we have structural MRI, diffusion MRI, and functional MRI. And then on the right, we have PET and EEG. And each of these axes give some attribute that if the point on this axis is far away from the origin, that indicates that this particular imaging modality, that's one of its strengths. So for example, structural MRI has great spatial resolution and is generally highly available in the clinic, whereas diffusion MRI also would have pretty good spatial resolution, but might be less available in the clinic, and so forth. So the idea is that we can leverage all of the strengths of each of these individual modalities, we can really get a better understanding of the brain, a more comprehensive understanding of the brain's structure and function. There have been some really amazing talks here at SMI on multimodal analysis, and so there's a lot of new methods that are available to integrate different types of imaging modalities in a single analysis, but we still oftentimes see in applied imaging analyses researchers just focusing on single modalities at a time and maybe relating these to phenotypes of interest. So I'm just giving one of course, many examples of a mass univariate analysis where this was a paper led by Ted Satterthwaite, where they were interested in looking at cerebral blood flow and how that changes throughout adolescence during neurodevelopment. And what they found was that in some particular regions in the brain, that males generally had average decreases in CBF throughout development, whereas the females tended to decrease in early adolescence, but then had this increase in later adolescence. So I picked this example because later in the talk, I am going to be focusing on some applications with respect to cerebral blood flow, which was what they were modeling here. So I tried to condense down to a few of the main advantages that I think the integration of multimodal imaging data can have. And I probably don't have to convince you all of these advantages, but the first is that we can potentially build better predictive models for example, predicting disease, so helping with diagnosing diseases, as well as monitoring progression of neurological and neuropsychiatric diseases. And then the second bullet point here is what I'm going to focus the majority of my time today on, which is the ability to characterize relationships between image modalities. So I'm going to try to convince you that that is something that is useful and of interest to our scientific understanding of the brain. And so then if we combine these first two advantages of multimodal data integration, the hope is that that will give us a more comprehensive understanding of how the brain is structured and functions normally, as well as being able to identify deviations from normal brain structure and function. So the goals of this work are to develop methods for estimating spatially varying relationships between different imaging modalities, and then apply these methods to problems in various areas. And I want to point out that this very much builds upon foundational work that was done by Simon van de Kar and co-authors, where they introduced this measure of local cortical coupling. So the idea of local cortical coupling was that they took, for an individual subject, the cortical thickness map on the surface, as well as a map of sulcal depth on the surface, and they looked at vertices within local neighborhoods centered around a particular vertex. They fit a locally weighted regression and extracted the slope of this weighted regression and deemed that slope estimate to be a measure of the coupling of these two surface measurements. So we thought it would be relatively straightforward to extend this idea of local coupling to volumetric space. And so that would enable us to study relationships between, for example, functional imaging modalities and maybe structural or another functional imaging modality that is also measured in volumetric space. And so the idea here is that we have two imaging modalities from an individual subject that are registered to the same space, and we just fit a locally weighted regression of using voxels that are within a neighborhood of a center voxel. So we move that neighborhood around the brain and then extract some feature of interest from this locally weighted regression. So to give you a sense of the model and our estimation strategy, I'm going to introduce some notation. We're going to assume that both images from each subject have been registered to a template space, and we don't necessarily have to register these things to a template space. We can calculate coupling within native space from two images from an individual, but we're going to assume that we're registering to a template space because maybe we're interested in downstream analyses that look at relating these coupling relationships to phenotypes of interest in a sample. Then we're going to let V denote voxel indices within a mask of interest. So in our applications, oftentimes we're interested in maybe studying relationships within the gray matter. And so if we had a mask of where the gray matter voxels were primarily located, then we could index the voxels within that gray matter mask. And then we're going to let yi and xi denote two vectorized images observed for subject I that are going to be of length capital V, so the number of voxels in the mask of interest. Then we need to specify a radius for which to calculate these locally weighted regressions. And so we're going to let little r denote our radius of choice, and then we're going to use a symmetric neighborhood of voxels. And so we'll let capital R denote the 2 times our choice of radius plus the center voxel cubed. That will be the number of voxels that we use to fit the locally weighted regressions. Next, we're going to let this S of V be a map that just extracts the voxel values from a particular neighborhood centered at voxel V, so that we can, if we apply this map S of V to yi and xi, that will give us the voxel values that we need within the local neighborhood to fit our weighted regression. So here's the model at voxel V for subject I, and if we let J index the voxels within the local neighborhood, then we specify this linear model that relates some outcome, the image modality that we deem to be interesting as the outcome image modality. We specify this linear model relating that outcome image modality to our independent variable image modality. We have this vector of errors that we assume to have mean 0, and then if we stack the errors together from the neighborhood, we also assume that the covariance matrix is equal to omega here, and we make a very restrictive assumption that this omega does not depend on location V or on subject I. So that it's constant within all of these neighborhoods and for each subject. So it's a very restrictive assumption that we really don't believe, but we do have some empirical results that I won't have time to present today that show that this is not such a, it doesn't do so poorly, in fact. And so with this model, we're interested in estimating these beta parameters, which are specific to each individual I and each location V. So we fit this model, which we refer to as our IMCO model, using weighted least squares. So how do we specify the weights for the weighted least squares procedure? We let D of J and V denote the Euclidean distance measured in millimeters between our center voxel V and a voxel J within the local neighborhood of V. So using that distance function, and we don't have to use Euclidean distance, but that is what we used in our applications so far. Then we specify the weight for voxel J by this function, which is just proportional to a Gaussian kernel that depends on that distance. And so what this does is it upweights voxels that are close to the center voxel V and downweights voxels that are further away. It also has this tuning parameter sigma squared, which we can use to control the smoothness of the estimated IMCO maps. So thinking back to the model misspecification, this setup corresponds to assuming that this omega covariance matrix is diagonal with an inverse equal to matrix W, where on the diagonal of W, we have the jth weight. So now if we define x tilde to be a column of ones for our intercept, and then our independent variable image modality, and then I'm going to drop dependence on V when I specify this beta hat IMCO here, just to make it easier to look at. This is our, of course, weighted least squares estimator for the IMCO parameters of interest at location V for subject I. So Ali did some great work using these IMCO estimated features, particularly or specifically, she used the IMCO slopes, so the beta 1 IV here, and she calculated these things for every pairwise combination of flare T1, PD, and T2 images that were collected from a cohort that had multiple sclerosis. So the idea here was that she showed that specifying a model to segment lesions using just these four modalities was outperformed when she additionally added in those IMCO features that quantified the relationships between each of these modalities. But what I want to spend most of my time talking about today is our application of the IMCO framework to try to study the coupling of cerebral blood flow and gray matter density throughout development. So in order to study these relationships, we were using data from the Philadelphia Neurodevelopmental Cohort, or the PNC. And to calculate gray matter density, we used the T1 weighted structural imaging from the PNC. And then to calculate cerebral blood flow, we were obtaining that from an ASL sequence collected as part of the PNC. However, we ran into this problem when we were studying the relationships between these specific modalities, which is that we know that blood flow in gray matter is much greater than blood flow in white matter. So when we're calculating the coupling of CBF and gray matter density near the boundaries of tissue classes, we know that this is going to be a mixture of the within tissue coupling of these modalities. And so this figure is just demonstrating that if we're in a neighborhood that is consisting of a mixture of gray matter and white matter voxels, the relationship between CBF and GMD for this particular individual looks much different than the relationship at a different voxel that contains primarily gray matter. So we had this idea, which is that we could use the IMCO framework that we've been developing to correct for this problem and to correct for partial volume effects in CBF, which is of interest to people that work in ASL and CBF. So partial voluming results from the finite resolution of imaging. So because voxels have a certain volume and they're not measuring things with infinite precision, we get voxels that span the white matter and the gray matter, or potentially the white matter and the CSF and so forth. And when that happens, the intensity within those voxels is a mixture of the intensity that is brought about by the properties of both of the tissue classes. So because this wasn't our original application, we kind of went back and did a literature review of methods for correcting for partial volume effects in CBF. And interestingly, we found that many of these methods use this idea of local regressions. And we found this method specifically by Algren et al. That fit a regression of each individual's CBF image on measures of gray matter and white matter. And importantly, they did not include an intercept in this model because these typically add up to be close to 1 in the areas of interest. They are also assuming that the contribution of CSF is 0 in this model. So they fit these regression models in local neighborhoods, but they do this slice-wise, and they also don't do any weighting. They just use ordinary least squares to fit this model. Once they fit this model across the brain, they extract the parameter that's associated with the gray matter term and say that that's their corrected version of CBF. So that's the CBF that would be measured in the gray matter portion of the voxel. So I argue that our IMCO model is similar to this, but improves upon this approach in several key ways, which is that, first of all, we're just regressing CBF on gray matter, and that we also have an intercept in this model, but that allows us to predict what CBF would be if a voxel had 100% gray matter. So we can get our corrected version of CBF very simply by taking the predicted value from this regression model. And then, as I mentioned before, when I introduced the IMCO framework, we fit this model in 3D neighborhoods, and we also use weighted least squares. So and we're referring to this particular application of IMCO as intra-subject locally adjusted CBF, or ILA CBF. So I just want to show some preliminary results here. We're still sort of working on this method, but in the top row, we have the average uncorrected CBF, so the average raw CBF across participants that had imaging in the PNC. The middle row here, and we've projected our results to the surface, as you can see here, but we did calculate these things in volumetric space. But this was just for visualization purposes. So what we see here, ILA CBF seems to increase the value of CBF throughout the brain, but retains some of this interesting variation that we are hopeful might be scientifically interesting and related to developmental effects. And then what we see is that the Algren method really overcorrects and oversmooths throughout the brain. And this is also noticeable when we look at each individual. If we bin or if we classify their voxels according to how much gray matter is in those voxels, and then take a look at the average CBF within those bins across subjects, we see that the raw CBF does indeed seem to have this partial voluming where for areas with low gray matter, it sort of underestimates what the CBF should be. ILA seems to correct this quite well in ranges of gray matter density that are lower, and then we see the overcorrection of the Algren method for a lot of these gray matter ranges. So in conclusion, we're developing this framework to be used to both generate additional features that might be useful for predictive models, like in the multiple sclerosis application, as well as to address partial volume effects in CBF images, as long as we have access to a gray matter density map for each individual that can be obtained from, say, a T1 structural weighted image. And then we're also able to use this framework to study spatially varying relationships between imaging modalities. And because the IMCO method is estimated at the subject level, we think that quantification of the relationship between imaging modalities might be useful as biomarkers, particularly if the actual modalities themselves considered in isolation aren't powerful enough as biomarkers. And then finally, we might be interested in downstream analyses where we relate the IMCO features to phenotypes of interest. This can be done using a two-step approach where we calculate each individual's IMCO image and then do, say, like a mass univariate analysis or whatever fancy method you want to use to estimate relationships in the sample across subjects. We're also currently extending the IMCO, the two-step approach, to be able to jointly estimate these things in a single-step estimation procedure. And we hope to develop inference procedures for that estimation approach as well. So with that, thanks very much for your attention, and I look forward to the floor discussion.